So, ladies and gentlemen, this is our first seminar this season. We were going to have one last week, but we got slowed down. We have a fairly full schedule. Next week there's no seminar because some of us are going to a conference, but from then on things are pretty much settled. Well, it gives me great pleasure to introduce Toby Berger, a former colleague of mine from Cornell, who is now, I suppose, Irwin Jacobs Professor of Electrical and Computer Engineering. Irwin Jacobs is who is probably the most famous now graduate of Cornell Hotel School, and is the president of Qualcomm. And Toby got his undergraduate degree at Yale and his graduate degree at Harvard, and he's going to talk to us on information and decision theory, explain why there are neural pulse trains. He will explain it.
Thank you, Fred.
He graduated from the hotel school, right?
No, he attended the hotel school. He graduated from electrical engineering. But he did spend his first 2 years at Cornell at the hotel school. But that isn't worth correcting. The only thing you said that's worth correcting is that one has to be very careful to say that this is not the Irwin Jacobs Professorship of Engineering, but the Irwin and Joan Jacobs Professorship. These things are sensitive. You have to be careful. As I said to some people earlier today, I tried to generate some sympathy for Irwin, who lost on paper $2 billion in the month of January, but nobody seems to. I get as much sympathy when I say that as I do when I complained when I went to Hawaii for a week to see the eclipse of the sun, that the clouds covered it up and I never saw it, and I've never gotten a grain of sympathy for that one either. Fortunately for Irwin, he has literally probably twice that many billions still left, and the stock is coming back. But we're not here to talk about Qualcomm today. We are here to talk about information and decision theory predicting the existence of neural pulse trains. And I have the right place and the right time. I'm starting with slide number 11. This being a consequence of when I gave a similar talk at UCSD about a month ago, Alon Orlitsky, I think it was Alon, said to me afterwards, well, why did you bother with slides 1 through 10? Why didn't you just start with what was interesting? So, and I had an emotional attachment to things on slides 1 through 10 because I'd been mulling those over in my brain for decades, but I guess thinking about it afterwards, I guess I decided that Alon was right. And there's always so many slides that even with those gone, I don't make it to the end anyway. So I'm going to say a few things first about differences between interorganism and intraorganism communications as I see them. So for 50 plus years, I have to change this again and make it 2000 now. Information theorists have restricted their inquiry, as it says here, almost exclusively to what I would call interorganism communications. And I put organism in quotes because the organism, for this slide at least, could be a machine as well as a living organism. And so organism A is trying to communicate something to organism B. We all know that there are such things as two-way channels, and sometimes you're trying to send stuff in the other direction as well, but we will ignore that totally today, although it's certainly present in the biological applications as well. So there's some given source. That organism A is seen as a source of information that I'm here designating as UL, and you have no control over that. And then you, as communication system designer, you design the encoder, which is sometimes sort of broken down into a coder and a modulator, but you design the stuff that goes between the source and the channel. The channel is given, you can't do anything with that, and you then design a decoder, and you feed that stuff to a given user or organism B. There are some things that characterize this problem. The nature of the interorganism communication with that block diagram has several things listed here by the bullets. Channel is given. This is usually assumed. Next thing that's almost exclusively assumed is that the channel behavior is independent of the source statistics. So the channel is going to do the same thing no matter what kind of source you stick on it. The next thing that comes out of studying the theory is that efficient use of the channel requires that you build complicated codes that have long delays and complex computations that have to be done to do the encoding, especially for the channel problem, and especially the decoding and channel decoding, or especially the encoding and the source problem. Those are computationally tough. And if you don't build these long delay codes with heavy computation, that in all but some very special problems, you perform suboptimally and don't do the best in source and or channel coding. The next thing is that there's some sort of criterion from the point of view of the user of this information about how good a job the system has done in encoding what mattered and getting it to him. And this source user fidelity criterion is usually relatively inflexible. And another thing that always happens in all the information theoretic interorganism problems is that the source and the user get together beforehand, and they establish and they exchange a set of coding rules. They have some sort of common language that they share, which may just really be, index numbers that represent a certain set of messages in the source. So this is message number 43, and it means the following. And similarly for channel coding, there's some kind of language, often simplified into the form of indices, that gets exchanged so that people have to do that before they can communicate. Well, not people, but these organisms, this source and user in this classical communication problem. Let me say a little bit more about what I meant by the channel is given. I mean, the most recent important example of that would be the sort of thing that happened in wireless communications. The FCC in this country and similar entities in other countries opened up some portion of the spectrum to send things in cellular, and then later another portion of spectrum for PCS, and they tell you, well, you have this many megahertz and such a band, and this is the maximum power you can use. You can't let more than the following power leak out into neighboring bands. You have the following interferers in there that we can't take away, and you have to figure out some way to cope with the fact that these scientific instruments and other things are generating stuff in your channel band. But it's yours, go use it. And then people go design stuff to use it. And I want to contrast that sort of situation with what happens in intra-organism communication, where I have some communication that is going on within a given real organism now, biological, biochemical organism. And there I feel that what happens is that the channel, if you want to talk about it, this is some sensing channel that's sensing either the state of some signal from the environment or perhaps the state of some internal signal, like how much salt or alcohol you have in your bloodstream at the moment. It adapts itself to the source that needs to be looked at. This happens both in a species ways and over the long term in a Darwinian fashion, that you adapt over the eons in such a way that this sensory channel that you have built is matched to the environment to extract from it efficiently what it is that matters to you. You might even say that that defines what it is that matters to you. And this also happens on a shorter time scale, where I don't have time to go into it right now, but things like with blood osmolality, the salt level in the blood, the signal that the salt level has gone up or down gets sensed by the hypothalamus, which produces a chemical called vasopressin, which it greatly amplifies a small change in the osmolality and pours that into the bloodstream through the, I get it right now, I think the pituitary gland. And it comes in there and it gets circulated around back to the heart. It comes in to the heart and it gets pumped out to all the veins and all the arteries. And all the arteries neglected except the renal artery, which is very interested, not the artery, but the kidney, when the stuff arrives there from the, when there's vasopressin there, this channel, which ordinarily, I do the kidney in a very simple binary model, let's see, for osmolality purposes, is having water molecules and salt molecules coming in, and it sets up a random transition between those two inputs and two outputs. The outputs may sound like they're not the same, they're not chemical, they're actually places, but information, which is in slides 1 through 10, information in living systems has to do with the time variations and the concentrations of chemicals in compartments. And so every piece of chemical concentration is indexed by two things, you know, what it is and where it is. And so the output in this thing is not chemicals, but where they go. So each water molecule either gets recirculated through the vein by being sent to the renal vein to go back, or else it gets excreted by being sent to the bladder to be excreted. And there's a complicated process of things that happen, but eventually it ends up in one of the other places. Likewise for the salt molecules. When this vasopressin signal comes in, that channel gets changed to change those transition probabilities, a control mechanism, and that's how this happens. And the signal itself is responsible for redefining the channel to solve the problem that needs to be dealt with. This is not the way we're used to thinking about things, but I think it's what happens. I believe that joint source channel coding is sufficiently performed by these systems at rather low delay by a very simple probabilistic transformations, more or less memoryless or close to it, without any coding as we usually think of it in information theory. I changed that recently from no coding to little coding. I guess maybe as time goes by I'll change it more and more and I'll give up this idea. But at the moment I feel that this is the way it is. That is to say, I don't think that all the stuff with all the beautiful convolutional and block codes and stuff that we learned about in information theory has anything to do whatsoever with how any human or other animal processes anything. I don't think that has a role to play. Now, that may sound like a bad thing for an information theorist to say, but we all know there are two kinds of information theorists. There are the probabilistic information theorists, and then there's algebraic information. So heck with those algebraic guys, that's not my thing anyway. So this is how I see it. I also feel that the source user fidelity criterion is rather flexible, especially in higher organisms with capable brains, and all you really need to do, this is a simplification, but to a large extent, all you need to do is if the same signal shows up, you got to get a reasonably high data rate that produces the same thing, more or less, to this high intelligence in the background there, and then you'll be able to figure out what to do, even if it looks very different from me than it does to you. And it's this business, you know, when I see green, do I see the same thing that you see when you see green, or do I see what you see when you see red? You know, who knows? But as long as you get the signal there consistently, to a large extent, it won't matter quite so much what it is. Now, obviously, if you get some kind of aberration in the visual system and you don't put glasses on, even though you consistently present the same thing, you can't decipher it. So I'm not trying to say that it's quite that simple, but it's to a large extent true. There's a lot of flexibility. And as I said already, no codes, no coding rules, no language. All right? Now, then comes the famous slide. I'll tell you why this slide is famous.
When you think of these power control schemes, you're not thinking of them as a channel adapting.
Power control things?
So if you had a wireless or a cellular situation where you change the...
Well, that of course becomes a question of, as always, unclear of where you draw the boundary between the part of the system that you designed and the part of the system that's the channel. I guess I'd say there that you designed that into the system, but you certainly have you have an adaptive strategy, I think. I don't think the channel changes, but the strategy for, but if you want to say that part of the channel is the power that you're allowed to use to send over it, which we certainly do that with analog channels. We, in order to fully describe them, we have to say what the power constraint is. And if you change the power constraint, maybe you've changed the channel. I don't know. Okay. This is a famous slide. It says, wait, what about DNA? Okay. The DNA is definitely a long block code, and it's in a nearly universal alphabet or language. It has intricate rules. It has lots of redundancy. Maybe this redundancy is used for error correction and so forth. And true, that's what I say. But DNA is used, it is used for certain intraorganism purposes that are very important, but DNA also has a fundamental role to play as an interorganism communication scheme, and therefore it is not subject to what it is that I'm talking about here. Now, you know, things like the DNA also is involved in having to do with gene expression and ontology and all that kind of stuff. Certainly I'd say that's within a certain organism. If I'm putting together a general theory of this, which I am, but I'm not talking about that general theory today. You need to be able to deal with that part. But the fact that DNA is a big, long block code with all kinds of rules in it doesn't, I think, in any way violate what I had to say. To a lesser extent, it's also taking place on a 25-year generation time scale, where the other things that I want to look at are on milliseconds or hours. But I think this is what's more fundamental, really, than that, is that it really is interorganism communication, or at least has an interorganism communication problem that it needs to address. Why is this slide famous? Well, this slide is really indeed 2 years old. And the first time that I showed this slide was in San Diego at the Salk Institute. I was in some postdoc at the Salk Institute, came over to UCSD EE department with some kind of information theory / biological question. And he found Jack Wolf and asked him this question. And Jack said, I don't know anything about that, but Toby Berger's thinking about things like that at Cornell. Why don't you contact him? So this guy sent me some email, and we exchanged email about his problem. And then there was an information theory workshop scheduled for that February in San Diego, just about 2 years ago. And I was going out there, and I told him that. He said, why don't you stop by the Salk Institute, give us a talk on whatever. I said, okay. So I got to the Salk Institute at 10 o'clock, and he showed me around. And then at noontime, I went to the seminar room. This was the first time I had ever dared to speak about anything that I did to actual biologically knowledgeable people. And I filed in at noontime, and the seminar lasted an hour, and then people went to lunch. So I start my big stack of slides up, and this one was not as near the top as it is now. And I get an interruption from somebody. It was actually a very distinguished gentleman sitting over on the right side of the room, and he asked me this question. And I tried my best to answer it, but I tried to dismiss it because I knew I had a lot of things I had to do. And then a little later, I get another question, rather antagonistic, from somebody sitting about where you are, and a younger fellow. And he says he didn't really see how what I'm talking about fits this, that, and the other biological thing. This seems to contradict that. And fortunately for me, Terry Sanowski, who works there and heads a group and understood what I was talking about and the biology and all that stuff, starts answering this question. So he and the other guy across the way, the three of them, are having this discussion about this question, which seemed to me like it lasted 15 minutes. It probably lasted 5 minutes, but that seemed to me like it was 15 minutes. And then I continued, and I got like partway through and then I got to this one. I put this one up, and the distinguished-looking gray-haired guy over there asked me another question, and he said, well, do you really think that that the redundancy in the DNA sequence is really known, that that's responsible for some kind of error-correcting coding capability? You know, I said, you know, well, it's not clear-cut. It's very controversial. Some people feel this is true, others less convinced. And then maybe a few more words. And then we went on with the talk, and the talk ended, and everybody filed out of the room except the postdoc who was had invited me, and I'm putting all my slides back together. And he said to me, do you know who that was who asked you that question? I said, oh, who was that? He says, that was Francis Crick. So I'm telling you, it's a DNA slide, and I'm saying, well, you know, DNA, this and that. So he still comes there. They say he, you know, comes to work about four or 5 days a week, works on things, sees what all the young people are working on, and so forth. All right, now I want to talk about this. I want to talk about something that I call a generalized Shannon-Blackwell Billiardball Channel, which I view as a mechanism for coding-free, or maybe I should now stick an insert in here, nearly coding-free, optimum biological information signaling. Okay. I hope I can justify that parenthetical description of this mathematical model. The classical Billiardball channel is binary input, binary output, and it has a storage of size 2. And what happens is that, you know, the input person devises some sequence of zeros and ones to stick into it of his choosing. And the channel does the following. At each time step, it's a discrete time channel, and at each time step, well, you can, where the boundary between one time step and the next one is can be chosen in different ways. But one way of doing it is to say that time step starts with the person from the output looking at the input at the store of the channel, or reaching into this grab bag of size 2 and randomly choosing out one of the two things that's in there and making that the next output, after which the person at the input sticks in a new symbol. It's called Billiardball Channel because the other way of looking at it is that that's not what happens, but the person at the input has white and black balls, which are the zeros and ones, and he has this cue stick, and he bangs either a white or a black ball in of his choosing into the thing, and it hits one of the two things that's in there and knocks it out. And then you do that over and over and over again. This was introduced because in the 50s, shortly after Shannon's original paper, people were interested in various channel models that did have memory that were simple. And this was a rather simple looking, at least easy to describe, model that had memory. In fact, it has infinite memory because the length of time that a given symbol stays inside the channel store is geometrically distributed and therefore has no bound on it. This thing has infinite memory, actually, both at the input and the output. So I want to talk about a generalized billiard ball channel. Instead of storing just two things, it's going to store what I call B things. I guess B is for Blackwell or billiard board or something, but I picked it B. And what's going to happen, you store B, and then this output mechanism does reach into the big grab bag of size B. By big now, I mean it usually has 1 million or more things in it, which I think of as chemical species. There are ions and molecules, stuff like that, and they're floating around in there. And you reach in and you pull one out. And the way I'm going to do it right now, you pull one out totally at random from all the ones that were there, no more or less likely to be the most recent one or the one that's been in there longest. That's probably not true because probably really things are sort of diffusing from this input to this output, and maybe something that's been in there a little longer might have a slightly higher probability of coming out than the next thing. But I'm interested mainly in synapses, and the dimensions of these synapses are very, very small in terms of the cross distance relative to the other distances, so it's not too bad an assumption, and I'm certainly going to make it. The alphabet is M-ary, though I'll reduce it to 2, as information theorists always do very soon, because it captures nonetheless the important aspects of, most of the important aspects of what's going on. So here's a Blackwell type mechanism for intercellular communication. I'm very interested in intercellular communication. So cell A over on the left side of the picture communicates something to cell B. I won't say that cell A wants to communicate something to cell B. That's anthropomorphizing cell A. I think it can't help communicating. It just does it. And so here's this setup where there's M different types of chemicals that might be emitted through the cell wall of cell A into the space between the two, and they're indexed from 0 to M-1, and they're floating around inside there having been emitted in the past. There's little b of them altogether. B is large. And then in the precise model, what happens is that, you pull one out and you stick one in, and then you pull one out and you stick one in. That's probably not what really happens. They probably come out and they go in and out at Poisson instance, but there's not too much difference from saying it. Just make it look as if it's ping pong in, out, in, and out. So that's what I do. And as I say, there's this diffusion problem. So this 5 that just came in is probably less likely to get picked next than something that had time to diffuse over near the cell wall of the other next cell. But I'll neglect that because really the picture should be squashed in such a way that this is very small and they're all in there. And it's important that there's lots of them. Very important. I mean, this is going to be a large B theory. But B is always large in practice. Okay. So this is the billiard ball channel with parameters little b and m for the size of the store and the size of the alphabet. But I'm only going to consider from now on m equal 2. But b is still large. So B is going to be 1 million or more, and we'll let the two symbols be represented by 0 and 1, but very frequently they are, ones might be sodium ions. That's the most important practical application. And what are zeros? Well, they're not sodium ions, or they're things that don't have a positive charge on them. There's several ways of looking at that. It's not too important right now, but let's just say that there are ones and there are zeros in there, okay? And here's the key question that I want to address. I'll move it up higher so everybody sees it well. Suppose that the density of ones in the channel store is P a priori. And now I decide, this is an I, a me type I, and now I decide to input capital N ones in a row. No zeros, just ones. All right, the next N time steps. How big does N have to be for someone who observes the next N outputs to determine reliably that I've been putting in all ones the last N time instance, as opposed to, say, putting in all zeros, or if you like, continuing to put in ones, IID probability P, like in accordance with the prior density. I don't really care too much which of these two things you pick as the alternative hypothesis. And when I envision this, P is going to be small and B is going to be large. What a typical number. B is 1,000,000 and P is 1/10 or maybe less. Sometimes in certain applications, much less. Okay. All right. Now here's the answer. Quite possibly the most useful thing or important thing I may have derived in my by now long and aging career. Here's the answer. N is equal to P to the 1/3, B to the 2/3. Okay, this is very important. Okay, I think it's very important. I'll try to say why it's important because I'll try to have some slides that address that. But first, let's see where it comes from. Well, here's this alleged answer. And the answer says that, that N is much, much less than B because if B is 1,000,000, then B to the 2/3 is only 10,000, and P can't be bigger than 1. So it's saying that N is small compared to B. So let's assume that that's true and see if this assumption is borne out. So throughout the N channel uses, the density of ones in the channel store lies someplace between, if I am putting in ones, lies someplace between P that it was to start with and P plus PB plus N divided by B. Because there was PB ones in there to start with, and I add in N more. Of course, some of these ones may get pulled out, but at most it gets up to PB plus N out of the total B that are in there. So the density of ones is always someplace between those two numbers. So it's between P and P plus N over B, and N over B is much, much less than 1 according to my assumption. So it doesn't change much. And that means that these Bernoulli trials of picking out ones or zeros are not exactly Bernoulli trials because there's a change in the density in the P value, but it's small. So that changes only slightly. So the standard deviation of the number of ones that's going to be observed at the output during these N channel uses is about N times P, 1 minus P square root, or more precisely, it's that thing times 1 plus something of the order of 1 over B. Now, let's assume right now that P is small, not terribly small, but just say less than 0.1. Well, then in that case, the standard deviation of the number of ones that come out in N trials when I'm putting ones in is about, so it's like root NP. Okay, that's what it's like. I'll put that slide over there. I may want to come back. Now let's take a look at the expected increase in the number of ones, output ones that are observed because I've been putting in, if I've been putting in ones. Well, there were PB ones that were in there in the channel a priori, and they, of course, have no effect on this increase because each one of those either will or will not come out in the next N times quite independently of what I'm putting in. So they're not of consequence. What's of consequence is the N ones that I've stuck in. What effect do they have? Okay, so let's look at them. The probability that the little nth inputted one is still in the channel after capital N outputs have emerged is, well, it's got chance 1 over B of being picked, and there's 1 minus 1 over B of not being picked. And if it's not, has to be not picked N minus N times in order to still be in there at the end of capital N. So that's the probability that it's still inside. And therefore, the expected increase in the number of output ones is given by this formula here. I mean, namely 1 minus that, which is the probability that it did come out. And the probability that the little nth one came out is the expected number, the expected value of how many came of the number that, if I add that up over from 1 to N, that's the expected value of the total number of things that, of ones, the new ones that I stuck in that made it to the output by this time. Is there any question about that? So now you just add this up, which I've done here. You get a capital N, and you do a geometric series sum, and you get this thing here, and then you expand it out because the denominator is very nice. There's a plus 1 and a minus 1, so you just have a minus 1 over B. It cancels the minus sign in the front. It comes out in the front as a B, and you expand this 1 minus thing over here. You get that 1 and this minus 1 to cancel those things out. And so you start getting all these terms, and you add them up. And the biggest term that you, of course, it's very important that this second term here is B times minus N over B, and it cancels with a minus sign the plus N that was in the front. So the third term, this one, is the first one that survives. And that term is N squared over 2B. All right. It's a very quick way to derive that result in a dirty way that isn't quite as careful. You just say, well, on the average, there were, let's see if I remember how to do this. There's sort of, I started with a density of P, and on average, I kind of increased it to halfway between nothing and n over b, which is the most I could have increased it. So sort of on the average, it's at n over 2b, and then for the expected number of things that come out in response to that, if I do it n times, and every one of them has this problem, is n squared over 2b. So if you don't want to be careful about it the way I was on the slide with the whole geometric sum, you still get that result, all right? So that's it. So the increase in the expected number of ones that come out is n squared over 2b to first order in this n over b that was small. Okay. And now all we have to do is to decide whether or not we can detect that. Well, we can detect that at the channel output reliably that I've been putting ones in during the last 10 uses if the expected increase in the number of ones exceeds the standard deviation of the number of ones that are going to come out. How reliably? Well, if you just set sigma, 1 sigma the way I did it here, then it isn't very reliable. But if you want to change the constant in the front and make it 3 sigma, you can make it more reliable. That's not terribly important. It's just stick some factor in the front. And actually, my friends tell me that individual neural pulses don't matter very much by themselves, and therefore, it's really probably not necessary to have a very low probability of error in deciding that there was a pulse when there wasn't, or that there was a pulse when there, well, that there was an infusion of sodium ions when there was, or there wasn't an infusion of sodium ions when in fact there was one. So, okay. So, so what I need is for n squared over 2b to be bigger than the root np, or in other words, I need n to be bigger than some constant times p to the 1/3, b to the 2/3, or right now, for one standard deviation, the constant looks like 2 to the 2/3, or 1.5. Now, so that constant isn't very important, and therefore the crucial result as advertised is that n is on the order of p to the 1/3, b to the 2/3. Okay, that's where I got that from. Now, consequences of this formula. Well, suppose that we did before that p is 0.01 and b is 10 to the 6th. Then a priori there are p times b, or 10 to the 5th, of these ones, or sodium ions, or whatever they are, in the store before this experiment starts. Okay. And if I use that formula, p to the 1/3, so 0.1 to the 1/3, 10/6 to the 2/3, I get 4641. Or even if I stick that 2 to the 2/3 number in, I get 7366. That's how many I need to make that happen. Tells us that an increase in the density of ones from 0.1, or 10%, to a little less than 11%, 0.107, even if you throw that in, is enough for you to make this detection reliably. You don't need to change the density very much at all in order to know that this has happened when b is large and p is small. This is very important, I think. Okay? Okay. We've changed the one's density inside the store by less than 1%, and yet we somehow have reliably detected this. Okay, when cell B is a neuron, as I said, the active detection occurs when Na plus ions increase in density enough to raise the potential on the cell wall enough to trigger one of these neural pulses, thereby detecting that this Na infusion has happened. I'm going to consider the detection event. The consequence of it is that some action potential gets set off. Now, I'm being a terribly simplistic description of what goes on here. There's a huge number of other things that have to do with vesicles and calcium ions and opening up ports that let things go through. And for those who are interested, I could talk about that afterwards. If you want to, you can look at this same thing as what you have to do to the calcium concentration in the vesicle in order to make this thing happen. And actually there, the prior concentration is much lower. The P is much lower, and the thing is even more sensitive. But we'll just look at it this way, okay? Yes?
Can you explain what is the biological importance of this capital N?
Say it again?
What is the biological importance of this?
Well, I'm going to try to do that in some slides that follow, yeah. Okay, definitely going to try to do that. All right. Now, I promise that the next one will start to do that, maybe even the end of this one. Now, next thing I want to say is that just detecting this infusion of sodium ions isn't good enough. You must be able to continue to detect it if indeed it continues to happen. And you must stop detecting it if it stops happening. Okay? So the problem is that this poses a problem because there are neurons that often pulse hundreds of times a second. Lots of them do that. And after 100 such detections, then the prior density of sodium ions under that model would have begin to approach 100%, in which case all the derivation that I gave you for how this thing is going to break down and become inapplicable. And also the time between detections would increase because you'd have to run it for a lot longer and it just wouldn't work very well. It's obvious when you think about it that some sort of reset mechanism is needed in order to continue to do these detections or non-detections reliably. And all this much I figured out from thinking about information and detection theory without looking, in fact studiously avoiding looking at anything physiological about how things work, because I didn't want to know how people said that things work. I wanted to try to figure out how things should work. But now I had a prediction. This is like August 7th or something like that. And on that date, I opened up the physiology book that my son-in-law had given me about a year before, and I read this. This is Schmidt and Thu's Human Physiology from page 9. Okay. It says the following. The action potential is based on a cycle of Na inflow into the cell and subsequent K plus outflow. Ion displacements occurring during the action potential are negligible in comparison with the high concentrations of the ion in the intra and extracellular spaces. The change in the membrane potential during the action potential requires very few sodium ions per square area. Of course, during the action potential, the inward sodium current serves not only to, well, I'll skip that sentence, doesn't matter. But I want to now talk about the part that I boldfaced. It's not boldfaced in the text. In fact, it's even in a footnote, very small. But even this total ion redistribution is quantitatively insignificant in view of the hundreds of thousands of ions in the adjacent volumes. A single action potential has essentially no effect on the intra or extracellular ion concentrations. It would take a large number of action potentials to increase the intracellular Na plus concentration appreciably. And such an increase is prevented by the Na plus pump, which was described the page before that. And the Na plus pump is the thing which, after you've had an action potential, there's a period of time when you can't have another one, and during that time the cell is doing many things, and it flushes out this whole thing and resets the prior sodium concentration to about where it was before the previous detection. In other words, it does exactly what the theory tells you has to happen. And that's just the way it is. Right? It fits almost about perfectly with what I felt should be the case. So another consequence is the following. Okay. The information carried by a neural pulse train in the peripheral nervous system sensory application, I would say, resides, well, that's not just I would say, it seems to reside entirely in the variation of the pulsing rate versus time, and not in the detailed shape of the voltage pulses, nor in the detailed time separation between any two successive pulses in the pulse train. This is all peripheral nervous system. This is not relevant to what happens inside the brain, but in the peripheral nervous system. This is the case, and this fact has been verified by extensive experimentation over decades. But if one believes in the theory put forth I did earlier, you don't have to do those experiments. You can see that it has to be that way, because the timing just has to do with the random business of what you're pulling out, whether you pull out ones or whether you pull out zeros, and you have to wait for this thing to happen. And the standard deviation is just a standard deviation. It doesn't tell you exactly how long this is going to happen, and the Poissonness of when they're coming in and out isn't perfect either. So the time at which the pulse happens, the precise time of that can't matter. And also the shape of the pulse doesn't matter. Now, sometimes there are neurons that serve more than two physiological purposes, and there may be one kind of influx of one kind of positive ion that may make this happen. In another case, it might be another one, and possibly the shapes of the pulses are different there, so that might code for what's happening. But if you look at a single function neuron, like many of them are, that's responding to the presence or absence of one particular thing, then the shape of the pulse isn't going to matter either. And all the experiments seem to say that that's indeed the case. And this is very important, because if this shape did matter or this time did matter, those are continuous variables into which you could conceivably code an infinite amount of information. But this is saying that that doesn't happen. And all that really matters is the the sort of variation in the mean number of pulses per second that allows you to track some more or less analog phenomenon that this thing is trying to keep track of. Some phenomena are not too analog. They have to do with relatively small densities of photons in the visual system. So I don't want to say it's necessarily an analog thing that you were tracking. I had one little thing at the very bottom of this slide, which was an additional consequence, which is that the capacity of this channel model, this Billiardball channel model, binary one, would by this thing certainly be something which is at least as big as the minus 2/3 power of B. So that takes about, because here's a way of sending one bit, yes or no, to this question if you make the two hypotheses equally likely. You could send a bit of information, takes B to the 2/3 uses of the channel. There's a P to the 1/3 factor in the front, but that doesn't change that too much as far as the, so it looks like the capacity is at least that big. So being an information theorist, the next thing you want to know as well is that how the capacity varies with B. This is not the kind of thing that information theorists usually do. They usually look for an expression, a formula for the capacity. And here what I'm trying to do is not to get a formula for the capacity, but to find how the capacity varies with the crucial parameter. I want to know what order of B it is. Of course, all this is not now in bits per second because this is in bits per channel use, and the length of time it takes for a channel use is another issue. And in fact, another thing that this sodium pump mechanism does is to speed things up. They actually often use up internal energy to make that thing pump faster so you can make these decisions faster. Sometimes it's worth that, and when it's worth it, the system does it. But anyway, but now I'm going to take off the whatever little biological hat I have and just talk as an information theorist about this channel model, because it's interesting. So in joint work with the incomparable Zhen Zhang, whom I'll be happy to talk about to anybody who wants to know about him, that doesn't already, we have determined that the capacity of this Billiardball channel is indeed of the form C being on the order of B to the minus 2/3, okay, for large B. The direct part of this result, that is that there are reliable communication systems that work at any rate that's smaller than B to the minus 2/3 will be shown below, and the converse will be roughly sketched. I don't know if I'll take time to sketch the converse. Now there's a question that I've been asked, which says since C decreases with B, why not just keep B small? And then you'll be able to send stuff at a lot higher data rate. Looks like a good question, but I think it misses the point. I think what that would do is that it would result in data transmission at an unnecessarily high rate, which is wasteful of energy. If B is equal to 1, for example, then of course the channel is lossless. You put in a 1, you get out of 1. You put in a 0, you get out of 0. There's nothing stored. Right? And it's non-permuting. If you make B large, that lowers the data rate. And what it does, of course, is to permute the inputs, which is, in fact, the whole idea. That's exactly what you want to do here. You want the inputs to come in because you don't care about the order of the inputs. You don't really care to preserve whether what I saw was 11 ones followed by a 0, followed by 10 ones, followed by 2 zeros, followed by. What I want to know is how many ones did I see and how many zeros did I see recently? I can permute them around in any way whatsoever, and there's no distinction from my point of view between these things, because there's no information carried by the actual order in which things come about. This is true of all of the biological models that I look at in the, when you look at them in the molecular level. Over the time constant of the phenomenon of interest, you can permute the underlying chemical symbols in any order whatsoever, and you don't change the information, because the information only resides in the density of chemicals in spaces. And it's not changed when you permute the order in which you look at them. So by doing that permutation, you preserve the concentration information, and you just make this thing much easier to run because it doesn't have to be a lossless, very tight little mechanism that runs very loosely. Okay. To really figure that out, you need to do more than I've done so far, which is to go and do an energy analysis of the whole thing for B equal 1 as opposed to B equal 10 to the 6th, and then look at the corresponding data rates and see if you're really using less energy per bit. My strong belief is that that's how it'll turn out. Well, actually, there's more to it than that I don't have time to go into. Okay. So here's some stuff about the direct theorem. First of all, I want to show that the capacity of this channel is at least as big as something that's order of B to the minus 2/3. And the reasoning that leads to the detection theory result with the P to the 1/3, B to the 2/3 isn't really adequate for purposes of a direct theorem in information theory in several respects. Two, anyway. The first one is that the information theory channel doesn't have a reset. All right, that's the first thing. And the second thing is that this is now I'm talking about not the physiological what happens thing that happens which has a reset, but this model which is just putting in ones and zeros and it never resets anything. And then the second thing is that at any fixed number of standard deviations, which you can obtain by increasing the constant in the front, the error probability is still positive, whereas capacity is supposed to be a rate for which you get arbitrarily small error probability. So you need to somehow cope with these things if you want to turn what I just did into an argument that shows that the capacity really is at least of order b to the minus 2/3. I think I'll skip what I have on the bottom there. So how do you do that? The key is to eliminate information bit sequences in which the number of ones ever either leads or lags the number of zeros so far. The running number of ones and zeros don't get very far apart from one another, so that you never allow a lead of ones over zeros or zeros over ones to get big, and you thereby keep the density of zeros and ones in the channel store very near a half all the time. And you want to send some ones, you send some ones for a while, then you send some zeros, but you don't let it get far. So here's a way to do that. Consider a 2k plus 1 state Markov chain that I show below, and this source or chain puts out a 1. Well, your source puts out a 1 every time that this chain steps to the right, and it puts out a 0 every time that this chain steps to the left. But you have reflecting states at the end, so whenever you get up to be k1s ahead of zeros, then you have to go back and put out a 0 next time, and likewise on the other side. So there's reflecting barriers. This chain has an entropy rate, which is obtained by working out steady state probabilities for the states, and it's a very simple thing to do. And when you do this, you find that the entropy of this thing is 1 minus twice the steady state probability of being in state k, because by symmetry that's the same as minus k. When you're in state k or state minus k, you have no entropy to the next symbol, and if you're in any of the others, there's entropy 1. So that's why it looks like that. And so I just need to look at the steady state probability piece of k of state k as k gets big to see what that looks like. And if you write down the usual equations for that sort of thing, you come out with a conclusion that 2 piece of k is equal to 1 over k. So 1 minus 2 pk, the entropy is 1 minus 1 over k, and it goes to 1 as k gets large. So if I build if I just, in the abstract, think about a Markov chain like this, putting out zeros and ones, and then I say that when I go to run this channel, the only thing that I'm ever going to let go in there are data sequences that this chain puts out, and not just any old sequences. So you can give in any epsilon, you can pick k large enough to make this 1 minus 1 over k bigger than 1 minus epsilon, and then you pick the standard deviation multiple in that argument that I had, so that this repetition code of putting in all ones for a while or all, well, you pick k big enough so that you can put them in long enough to get the standard deviation multiple to be large enough to make your error probability less than some 1 minus epsilon. And so by adjusting it like this, you can see to it that without resets, you can still send with this mechanism at any rate which is close to b to the minus 2/3. So you get that. And then in order to complete the proof that that's how the capacity really behaves, you need to prove the converse, which is due to Joan. And I won't go through the equations on the bottom of this, but I'd like to try to tell you what in my view, monumental piece of information theory it is that he was able to do this thing. Okay. Okay, so let SI be the channel state, that is the number of ones in the channel store between the insertion of the ith input and the extraction of the ith output. What you need to do is to upper bound the mutual information rate between n successive inputs and outputs conditional on some initial state by a quantity that's order of b to the minus 2/3. So you show that it can't get bigger than something that's b to the minus 2/3. And one of the things that has to happen here is that you cannot assume that the input is a stationary process. And in fact, the input that I used to get the positive result wasn't a stationary process, because I put in all ones for a while, and then I put in all zeros for a while. So somehow you have to run through an argument here that allows this thing to work for every kind of process whatsoever you could possibly stick in there without the stationary assumption, all the ones that we usually like to make, and somehow bound this information rate. And let me just suffice it to say that he has a way of doing that. And he made that happen. And so he came out with the conclusion that you can't get it to be more than b to the minus 2/3. There's, I'll probably stop showing slides at this point and say a few words about the evolution of that result. When I talked about this at the information theory workshop in Greece last June, July, at that point, all I had was a bound that showed that the capacity was at least 1/b, which of course is much worse than b to, it's very important than b to the minus 2/3. When you look at the numbers that I put in there, it makes it 100 times bigger. So it's very important to be able to do better than 1/b. Bruce Hayek was sitting in the audience and he said, after listening to the talk, I think I can see how to modify that to get log b/b. And he told me some things about how to do that. And it sounded like it made sense. And then I drove with Joan from Metsovo, Greece, down to Athens, and we talked about this problem. And by the time that we got to Athens, it was b to the minus 2/3. And then about a month later, he proved this thing that showed that it can't be better. So we really do know how it works. I think I'll stop there and not, since time is running out, not try to put on here what would have been slides 1 through 10 having to do with sort of a general theory of information theory and of information in living systems, what information means in living systems, how you tie this into chemical thermodynamics and the connections between rate distortion and chemical equilibrium theory, which I really started looking at 30 years ago and put aside for 27 years before I came back to it. But I'll not do that. I'll stop here. Okay? Questions? What time? Do we have time for questions? Is the first thing?
Yeah, we do have time.
All right, all right. I actually finished in time. It's a good idea to get rid of slides 1 through 10. I finished in time. Yeah.
Do you have any details to use this BB channel to describe how cells communicate with each other?
I have trouble hearing you from back there. It's partly you, and it's more me. I don't hear as well as I did. Yeah.
The main idea here is to use this BB channel model to model how cells communicate.
Yes, I believe the following, that at least peripheral neural activity, neural activity works in the following way, that you set up one of these Billiardball Channel models with, in general, not just 2, but m different types of chemicals in it. And then you you sort of pour through it in the way in which I did there. And I won't say there's no coding, because there was some coding. But it's a very simple coding. It's a repetition code, the one I gave you there. You put a near repetition code in, but you don't have to do it. It just has to do with the increase or lack of increase of certain chemical species. If they go up, then you don't have to really be putting in all ones. All you really need to be doing is to be putting in either more ones than you did before or less, or it's ones at some sort of mean density, and they then take longer to respond. But the idea is that these chemicals are coming along at the input, and you're trying to track their densities. And this kind of channel allows you to do that in what I believe is a very information efficient way, because what it does is to permute the order in which the symbols arrive so as to preserve for you only the density of the symbols over the length. Well, it depends on how frequently you need to read out the result, so to speak. I mean, you're looking at it all the time, but see, do you need to respond quickly to small changes in the density with this, or do you not? Would you rather wait a long time and have an accurate estimate but not get it often? Well, the same mechanism does both of those things and everything in between as efficiently as you can, in the sense that I believe that the reconstruction of the data Well, all you have to do is change B. If you change B, you change the data rate, and you can either get an accurate, for a small b, you can get an accurate reconstruction of something on the time scale that you care about at a high data rate. Or with a larger b, you can get a less accurate a reconstruction on a longer time scale, which is more accurate. I guess that's the way to say it. So you can trade these things off against one another, and I feel that in a rate distortion sense, this thing is near optimum all the time with no coding. In that it preserves, well, I have to tell you what it is that I'm trying to preserve. What I'm trying to do is I set up a window of a certain width, which is appropriate duration, which is appropriate to the application at hand. So in this blood osmolality thing, maybe it's minutes. And other things like vision, it might be milliseconds. So it depends on the application, but there's some window. You set this window up, and your job during that window, as that window slides along, is to preserve for the user, as accurately as possible, the time variations in the concentration of each chemical in the window. So you have a fidelity criterion, and the fidelity criterion is what we would refer to in information theory as a block distortion measure. And it slides along, and what you're doing is you're looking at the empirical distribution of the chemicals of the input in this window. And the idea is to get that to the other side with some accuracy. All right. But sometimes you want a small window, and sometimes you want a large window. And the same mechanism, I claim, does both of those things very well. In the small window, it gives you a large distortion, but you get the result in a small window so you can react quickly, but you don't have as accurate an input. In the large window, you react more slowly, but you have very accurate data. And both of these things are done simply by pouring the data into this mechanism, and pulling it out the other side, which permutes all the stuff. And you don't care about that permutation, provided that it happens on a time scale which is still no bigger than your window. So that you, maybe I shouldn't say any more, but that's the idea behind it, that I do actually, in fact, believe that the generalization of that mechanism to a larger number of chemicals at once is a very efficient way for tracking the time variations and the densities of chemicals, not the order in which they arrive, which doesn't matter, but the densities of them, at low expenditure of energy without any coding.
But these results are theoretical results. Have they been verified in...
I'm going to come closer again to hear what you're saying.
These results are theoretical results. Now, do we know
Yes.
whether two cells, even single cells,
Don't.
communicate this way?
I have no idea. Well, I don't know. I mean, I was like, my intuition makes me strongly believe that it's true, but I don't know. I'm not backed up by any results.
Is there any biological evidence that some macroscopic measurements agree with the formulas that you derived?
No, I guess I'd say the only thing I really have is some sort of a description there that seems to say that that's the way. From what I showed you there. No, I don't have. There's no quantitative measurements that I can tell you about that would. They may exist, but I don't know about them. And I don't think anybody took measurements with the intent of trying to verify or disprove this kind of relationship. I don't know. Yeah. Yes?
When you describe this store of ions, are you talking about the ionic composition of the synapse, of the chemicals that are in the synapse?
Well, the way I drew it there, I made it look that way. But actually, depends on whether you're, I think that's a pretty good model if what you're talking about is cell A not being a neural cell and cell B being a neural cell. Then I guess I'd say that that's what I'm talking about. But a lot of times, of course, you're talking about one neuron transmitting information to another neuron through that kind of neural synapse. Then, of course, the information that's coming in is already coming in the form of some sort of a neural pulse that's propagating down the axon of cell A. And as that happens, that electrical thing going by does indeed, in fact, result in this inflow of sodium ions and outflow of potassium ions a little later that comes through there. So that same kind of interchange is taking place, and it does eventually trigger at right near the synapse from this little vesicle that stores the things, this calcium things go in there, open up that channel and make it happen. So the mechanism is more complicated when you go from one neuron to another, but I think it's still governed by the same sort of considerations, which might be better applied to the calcium concentration in this vesicle than to the inflow and outflow of the sodium and potassium. But you're getting beyond the level where I understand it well. I've told you almost everything I know about that in my answer.
Because, I mean, the inflow and outflow of potassium and sodium, I think that describes a propagating action potential. But
right.
when a spike is transmitted from one neuron to another, then the chemicals are very different.
The chemicals that do that are different. There are neurotransmitters that are released. This calcium thing builds up in this vesicle. It opens up certain things and sends neurotransmitters. And the same sort of thing, I believe, will be true there. These neurotransmitters are chemicals, and you have to get enough of them to come on the cell wall to build up the potential. The same idea, I think, would apply, but it wouldn't be sodium. And the way it is, I think, when some muscle cell is trying to get some neuron to stick to, or some ganglion type thing in the eye is trying to get some neuron to be the first neuron in the chain to send something. So it depends on which of these types of things you're looking at. I was definitely, the way I said it, it's more like a model of muscle to neuron or ganglion to neuron, but I think if you get into this release of neurotransmitter and how much neurotransmitter has to build up on the cell wall of the second neuron to make it fire, I think you'll get much the same sort of conclusions.
I have two questions. The first is, do you have a sense for what leakiness would do to the billiard ball channel?
Do I have a sense of what?
Of what leakiness? Everything is preserved from the input to the output. So if the system had some state that it was, if cells restore themselves to some sort of resting state, if the system had a state, and so it was losing things to restore it.
So instead of they're coming in and all being taken out eventually, some of them are going off in other directions, which probably does happen in that matrix of stuff. There's lots of cells in the matrix. And, I haven't looked at that, but I suppose it wouldn't matter until there was a large leakage. If there's a large leakage, then at some point it would seems it would have to become very detrimental. Yeah.
And the second question. Sorry.
Yeah, no, you had two, right? Yeah, okay. Since the first one was short, you can ask the second one. Yeah.
I get the sense that this channel overall does something like a low-pass filtering, but maybe you can enlighten me.
I think your sense is right. I don't know that I would characterize it in quite that way, but it is maybe what I would say is that there's some sort of analog signal which has to do with the time variation in the concentration of some chemical, and you're trying to digitize this in essence with something which is not unlike a delta modulator or a sigma delta type of device, which is putting out lots of little pulses in response to what's happening. So I think I'd probably characterize it more like that it's some kind of a tracking, well, it's not a delta modulator because it has feedback in it. I don't see any feedback here, but it's some kind of a thing that's just more like an A to D converter type of way of running account. But then it has this property that depending on the size of the store, it permutes things. And that you could look at as a low-pass filter because it's only interested in that slower time scale and not really interested on what's happening at the level of the statistical mechanical level of the individual molecules, so that it's something of a, well, some people have said to me, well, that's what you're doing, and why don't you just look at the analog signals and forget about all this stuff with the molecules and so forth. Maybe that's something like the spirit of your question, so you don't have to bother. What's the point in looking at that? I was asked that once, and I guess my response to it is and I'm not saying you asked this question, but my response to that question is that's sort of like arguing, since we have thermodynamics and thermodynamics works, why look at statistical mechanics? But I think it's nice to have a description at the molecular level of what's going on that when you aggregate it makes sense so that it agrees with what you're observing in a more continuous kind of an environment. I don't know if that's if it answers your question, and it went off on something of a tangent, but yeah, there is something low-pass filter like that that's happening, but it's not quite a classical low-pass filter.
Yeah, I had a question about the BT channel, and I'm afraid this will have to be the last question. So anyway, my question was that when you did the forward part of the proof, this is a channel without any reset, you used this 2k plus 1 state Markov chain and used its output as what you tried to assemble.
Right, because now I'm allowed to pick the input from any way I want to.
Is that the capacity achieving process distribution?
Well, I mean, capacity achieving, it's capacity, it's order of B. If you look at how the capacity behaves as a function of B, it achieves anything, B to the minus anything bigger than 2/3.
Okay.
B to the minus parentheses 2/3 plus delta can be
Because
achieved
it
by
wasn't
this.
quite there because the way you chose the process depended on the epsilon that you wanted to beat. So you chose a Markov process.
That's right, yeah, but that's all I have to do. I just have to show you that I can build a code
Right.
and an input process that will achieve this mutual
Right.
information rate that will, you specify some epsilon you want and some, I say I can pick a rate, you give me a delta and no matter how small it is, I can send information at rate B to the minus parentheses 2/3 plus delta with an error probability which is less than epsilon. To do that, I need to know the epsilon and the delta. Then I have to pick this k big enough to make that work. But that's solely, I think, that you're expected to do in order to prove that
Okay,
that the capacity is order of B to the minus
would it be
2/3.
that the eventual, for a fixed B, would then a infinite Markov chain be the right distribution? Because after all, I'm just thinking in terms of if I didn't have an epsilon, if I just wanted to state something about the distribution which maximizes the mutual information for the channel that you sent it out. I wasn't clear about what role.
Yeah. Well, if I send that boundary off to infinity, then you get all sequences. And I can't allow all sequences. So I don't know. I guess I don't know if there's anything where I can make the process that gets as close as one specifies to the rate at a small error probability as wanted. Whether I can do that sort of uniformly in the delta that's near 2/3 and the epsilon that's supposed to be achieved by fixing some process. I don't know how to do that at the moment. I don't know if it could be done.
All right, thank you very much.